My name is Alene Abdulian, and I'm a high school student at Marlborough School in Los Angeles, and I've been working in the University of Southern California's Valero Lab under my mentor Hissam Azadju. The goal of this study was to explore the neural dynamics in macaque monkeys when executing freestyle hand movements to better understand neuromuscular function for potential use in brain-computer interfaces, or BCI, applications. BCIs are computer systems that can decode brain signals into commands and can be used to create life-changing solutions for those experiencing motor impairment due to neurological diseases like stroke or traumatic brain injury by eliminating the need for muscle movement when carrying out tasks. We worked in collaboration with Kazuhiko Seki, a PI at the National Center of Neurology and Psychiatry, who supplied us with a data set of the recorded electrocorticography, or ECOG signals in the macaque monkeys. Subdural nodes were implanted on the monkey's supplementary motor area, the M1 area in the motor cortex, and S1 area in the sensory cortex. The monkey was prompted to pick up a small morsel of food, and ECOG data was recorded when the monkey reached for the morsel and brought the morsel back to its mouth. In data pre-processing, filters were applied using MATLAB's Filter Designer app. ECOG signals were bandpass filtered from 3 Hz to 100 Hz to isolate signals in the alpha band through gamma band, and a notch filter was also applied at 60 Hz to remove the oscillations produced by alternating currents. We then downsampled the signals from 1,375 Hz to 275 Hz. The pre-processed data set was then passed through an autoencoder, a type of neural network designed for self-supervised learning that can take high-dimensional data and extract lower-dimension representations while reducing noise and preserving variation. We encoded the 80-dimension ECOG signals into a 16-dimension latent representation. Principal component analysis, or PCA, was then used to visualize the reconstructed signals in 3 dimensions, and a Savitsky-Golay filter was used to smooth the neural dynamics. The autoencoder successfully extracted the neural dynamics and was able to reconstruct the original signals with minimal loss, as seen in Figure 1.b. The trajectories of the neural dynamics during reach and retrieval were clearly different and occupied separate regions when plotted in a 3D plane, as seen in Figure 1.a, displaying that the neural patterns during reach and retrieval are distinct. This distinction can be employed in decoding the motor function targeted by the brain and creates a foundation for the development of BCI to aid those experiencing motor impairment due to neurological diseases. However, some limitations to this study are the simplicity of the task, as reaching and retrieval are only 2 examples of hundreds of functions the arm and hand can perform, and the invasive nature of ECOG might make these results a less convenient option for BCI. As a proof of principle, we showed that neural activity patterns associated with executing reach and retrieval movement in macaque monkeys are separable, and an autoencoder architecture can successfully extract such patterns. Though we did not perform a synergy analysis, the difference in neural dynamics suggests that the reach and retrieval actions in the macaque monkey utilize 2 separate muscle synergies. A possible next step could be performing a synergy analysis to support this assertion, using electromyography, or EMG data collected during this task. Thank you for listening, and if you have any questions, feel free to reach out to me